Today I am going to demonstrate the tool NAP, the Network Analysis Profiler. This is a tool for topological analysis of networks, and also it compares between two different networks. So I am going to demonstrate the new version, that is version 2.0, that is recently released. The original version is released in the year 2017. So this is NAP version 2.0, and first is we need to upload the network. So I am going to work on these 15 set of genes. These are basically associated with innate immunity, and whenever there is a virus attacks a host, the viral proteins interact with these 15 hub proteins, and these proteins were used to study drug targets against SARS coronavirus. So what we need here is to upload network file, that is in tab delimited format. So I created the network file. Like some of the proteins or genes that we saw in this are converted into network, and this is the target source, and this is the target, and these represent the weight of the edges. So this is another such file. These are the proteins that are present in this list or interact with the proteins of this list. So I am going to upload those. Set 1 is weighted, and let's name the network 1. Yeah. So this is how the set of gene and the interactions are tabulated with the source and the target and the weight. This is the uploaded data, and they convert it into 2D view. So this is how the first set of genes looks like. You can see STAT1 has the most dense network, and we can have different layouts according to our convenience. Maybe random for this. And after that, we also have this feature of 3D visualization. This is STAT1. So in the same way, we can add multiple networks. Let's go for GZ2, weighted, naming it network 2. We have to add it. And another file, GZ3. Naming it network 3 and adding it. So we can visualize different networks in this way. So go for the circle view. This is the second network, and this is the third network. After this, you can select different nodes and see their path. Like this is the OS to path. The next feature this tool provides is the topology. We go for the topology analysis. So these are the different parameters on which the topology is analyzed. For network 3, there are 146 edges and 16 nodes. This is the diameter and this is the radius, the minimum eccentricity. There are 0 self loops, and it also tabulates the average eccentricity and centralized betweenness and degree. This can be more clearly visualized in plot format. We can also compare these parameters between different networks. So green represents network 1, orange represents network 2, and purple network 3. So we can see that network 3 has the maximum number of edges and also nodes. The diameter of network 2 is the maximum among the 3 networks, and all the 3 networks have the same radius, that is 2, that is the minimum eccentricity or the smallest longest path between any 2 nodes. This is the clustering coefficient. You can see that network 2 and 3 have similar clustering coefficient. That means the neighbors of the nodes are more connected in network 2 and 3 than in network 1. This is the average eccentricity and centralized betweenness and average number of neighbors. The nodes have maximum neighbors in network 1. This is the centralized degree. So this way we can compare different network topology. After this, we can increase the size of the graph. After this, we can go for ranking. So what this option basically does in this tool is it ranks the particular protein or the node with the maximum value of a particular parameter. For example, if we go for centralized degree, you can see that for network 1, IFIH1 has the maximum degree. So it is the most connected node, and MT2 is the least. So this is how we can see that. We can also see the plot of a particular parameter of a particular network. This is the histogram of centralized degree. What we can do also is that we can compare between different parameters. If we select all this, a matrix of graphs is formed where the upper triangle shows the exact correlation score between any 2 parameters. For example, centralized degree and clustering coefficient has a correlation score of 0.9, that is, it's highly correlated, these 2 values. So basically what this means is that the more a particular node is connected to different nodes, the more are their neighbors well connected. These marks are these codes are also accompanied with these star marks. So the more number of stars, the more is this value significant, the more is the correlation. As we can see that clustering coefficient and eccentricity has a correlation score of 0.5, which is not at all a good correlation score, and this does not have any star mark. And the lower half of the triangle shows the scatter plot between the two. So if you are talking about centralized degree and correlation coefficient, this is the scatter plot. You can see this is correlated, like the score is almost 1, the graph is almost at a 45-degree angle, and this is the least correlation score between centralized betweenness and eccentricity. So the graph is also not a correlation graph. So this is how we can compare different parameters, and this can be done for different networks, and how the value of these networks are correlated. This is basically a regression line. We can erase that also. The last part of this tool NAP is intersection. It shows an output something like this. This shows that if there are any common nodes between any two networks. So let's select, suppose the first network is network 1 and the second is network 3 for demonstration purpose. You can see that edge intersection is 18.6% between these two networks. That means the source, suppose for example ATGC is the source and this is the target. Both the networks has this particular source and target edge. Likewise, there is no intersections. The node intersection is 26% between network 1 and network 3, and these are the 5 nodes that are common in the two networks. You can see a 2D format of this, and the 3D format produces more clarity. Yeah, so the red represents one network and the blue nodes represent a different network, and these yellow nodes are the common nodes between these network 1 and network 2. So these yellow nodes, also there are edge intersection because these particular part represent edge intersection, and this kind of edge is present in both network 1 and network 2. Similarly, we can produce different intersection. This is the intersection between network 2 and network 3, and you can see that the network intersection is more between network 2 and network 3 than what we saw previously between network 1 and network 3. This is more denser. Yeah, it increased from 18% to 30% edge intersection, and the node previously was 26 and this 36%. So there is more intersection between network 2 and network 3. So this is the final part of this tool, NAP, Network Analysis Profiler, and with this I conclude my demonstration. Thank you.